randomness or inhomogeneity, but there'll always be perfect orientation control. And that's done through the biosynthesis of the complexes. So energy flows from these LH2 complexes through an energy transfer process, eventually makes it into this reaction center where the electron transfer process happens. And this just shows sort of schematically the kinetics and pathway of this process. And this is one of the LH2 complexes, and I didn't show all the details of the molecular structures. There are two types of chromophores here. There's a very densely packed ring of chlorophylls that have absorption around 850 nanometers, and then there's a more monomeric ring that has absorption around 800 nanometers. And if you selectively excite into this 800 nanometer pigment, these act more or less like single molecules. You may occasionally get transfer around this ring, but mostly they'll just transfer to this more densely packed ring. Here you have very rapid energy delocalization around this ring. Really can't specify that just one of the molecules has been excited. Then over a period of a few picoseconds, that energy will migrate from this LH2 complex into the LH1 complex. And then again, you have a very rapid delocalization around the periphery of this, and then a much slower transfer, energy transfer into the reaction center, which is where the electron transfer process happens. And once that energy is transferred into the reaction center, it tends to not come back out very much. Probably 80% of the time it will be quenched by electron transfer, and the excitation will not re-escape to the trap. That's not true in all systems. In some systems, the energy can re-escape back to the antenna and go off and migrate around again and then eventually come back. But ultimately, in most cases, it is trapped by electron transfer processes at these reaction centers. Okay, so that's perhaps the best understood system in terms of structural information, spectroscopy. I didn't really show any of the primary data on that, but there's a wealth of time-resolved femtosecond, picosecond spectroscopy. And there's been a lot of theoretical analysis on those systems to try to really get a fairly deep understanding of them. The next system I want to talk about is the Photosystem I from cyanobacteria, and this is really a monster of a complex. In cyanobacteria, there are actually three separate Photosystem I complexes that form a trimer, which you can see here. Has a total mass of about 1 million Daltons, and it has about 300 chlorophyll molecules that are associated with it. Really a tremendously large and complex system, and this just shows it in several different views. A side view from the membrane. What we were seeing before was sort of a top view. Here now is just looking at one of the monomers of that trimer. Again, from a top view, looking at the two main parts of the protein complex, and there are multiple protein subunits here. I've stripped away a lot of that kind of complexity to get down to the essentials. Lots of chlorophylls. This just shows the chlorophylls without any protein. You can see this large. There's sort of two layers of chlorophylls, each surrounding this central domain, which is this electron transfer pathway, and electrons go along these two different arms of the electron transfer system, and eventually come to these iron sulfur centers and go out and reduce carbon dioxide. And so you have this large number of pigments acting as the antenna here, feeding the energy into this central complex, which does the electron transfer process. And you can imagine there's a lot of complexity in terms of the spectral inhomogeneity of these different pigments. They're not all identical in terms of spectral properties. There are a few of these pigments that have very low energy properties, so-called red pigments, and they have some important influences on the process. And so there's a lot of interesting stuff to be done with those. And we and a lot of other people have done quite a bit of work in terms of probing this system with ultra-fast spectroscopy, and of course all the structural work that we didn't do, but other people have done and have provided a structural basis for interpreting a lot of this work. But you can generally describe the excited state dynamics of these systems in terms of three fairly well-resolved lifetime components. You see a component of about 25 to 50 or so picoseconds that represents the overall trapping of the energy in the reaction center by electron transfer. So you give a short pulse of light, and then by 50 picoseconds or so, all of that energy has been converted into charge-separated states. Prior to that, you see these two separate phases. There's a very ultra-fast phase, which we interpret as terms of just local rearrangements within some of these nearby pigments in the sort of sub-picosecond range. And then there's a very clear few picosecond, typically 3 picosecond phase that represents the redistribution of energy into these low energy or red pigments, and that are the pigments from which the energy is transferred into the electron transfer chain. So this sort of the typical dynamics that you see in this system. But under some conditions, it gets even more complex. This is now a cyanobacterium that's grown under iron stress conditions. If you go out into the South Pacific Ocean, all of the organisms that live there are iron stressed. It's the limiting nutrient. And in fact, people have dumped 55-gallon drums of iron salts into the ocean. And then from a satellite, you can actually then see this tremendous bloom of algae and cyanobacteria that are responding to this increase in the amount of iron. And so in nature, these things are always under iron-stressed conditions. And they respond to this by adding an extra layer. The trimeric complex I showed before is just this green part in the middle. They then add an extra part around it, and it practically doubles the, not quite, but almost doubles the size of the antenna, because each of these 18 different complexes that are added in around the outside contain roughly 12 or so chlorophyll molecules. You add that to the ones that are in the trimer. And so what this allows this organism to do is to make fewer complexes. Each one of these Photosystem I complexes, trimeric complexes, uses 36 iron atoms. And so they're very iron-hungry systems. And so by making a larger antenna, in a sense, you're allowing it to use less iron per system. And this is, you can actually visualize these things, and that's how they were first discovered, was to see these in single particle electron microscope analysis. And this is this supercomplex formed under low iron conditions, and this is the normal trimer formed under iron replete conditions. And you can actually break down, there's slightly different absorption properties of the external ring, which is shown here in green, and the core shown in blue. And we can do at least partial selective excitation of this so that we can selectively excite this outer ring and then watch the energy as it flows around that ring and into the center of the complex and becomes trapped. And we can do various different wavelengths of excitation as well. And this is a little bit of a complicated figure in that these are different spectra. So this is basically a whole data set acquired on the systems without the extra ring, and then subtracting from that subtracting from that a second data set that's acquired with the complexes that have the extra ring. And so we're looking basically at the difference between these two systems, which hopefully reveals the effect of this extra ring of antennas. And this just shows some time resolved femtosecond absorption transients here at spectra at different delay times. And what you see is at very short times you have this extra excitation density that's in this shorter wavelength antenna, which is that outer ring. And then over time that decays away, and it looks more like the system did before the without the extra ring on there. And you can deconvolve this, analyze this in terms of what are called decay-associated spectra. So these are the different spectra associated with a particular kinetic component. We see a very fast component, again, which we interpret in terms of energy transfer within these clusters in the outer ring, and then some middle time components in which we interpret this in terms of equilibration and energy transfer into the center of the system. This is a very complex system, and it's not possible to really get down to a very, very detailed understanding of the energy transfer in it, but at least we can get a fairly overall picture of it. Okay, so now I want to turn to what I'm going to spend most time on, and these are the chlorosome antenna systems found in green sulfur bacteria. And these are true nanoscale devices, and this antenna complex shown schematically here, and here's some electron micrographs showing these sort of cigar-shaped structures which are pressed, stuck to the cytoplasmic side of the membrane. They're about 200 nanometers in length and contain roughly 200,000 molecules of chlorophyll-type pigments. So they form an efficient and ultra-fast energy collection system. We've done a lot of work doing selective excitation of these pigments in this complex and then watching the flow of energy through various intermediates. I'll tell you a little bit about some of these, and ultimately getting to the reaction center complex, which is embedded in the membrane, shown here by these lines. The thing that's most interesting about this complex is it represents Mother Nature's solution to the lowest possible intensity of light that can sustain life, sustain photosynthesis. And you can actually calculate that under some conditions, these things will absorb one photon every 8 hours per pigment. So these things are extremely low light adapted complexes, and we'll see some examples of organisms that they live in. And the other thing that's interesting about them is they have unique pigment organization, and I'll show you a little bit of structure of how this works. Pigment oligomers instead of the pigment proteins that we've seen in these other systems. And so they represent a simpler assembly process that will largely self-assemble, and we can make models of these. And I think they're at least interesting possibilities in terms of something that an artificial system could be built around. And finally, at the end, if I have enough time, I'll tell you a little bit about some of the redox activated modulation. If these things get too much light, they can fry themselves pretty quickly, and so they have multiple mechanisms of protecting themselves against that overexcitation, and we've done some work to try to understand that. This is an electron tomography picture of a dividing cell of these things, and these little jelly beans represent these chlorosome complexes, and each cell has roughly 100 of these complexes stuck to the membrane of the cell and transmitting the energy into the reaction centers, which are within the cell membrane. I mentioned these were the lowest known light intensity collection devices, and it turned out that through a long story, which I won't tell you today, we went down and looked for these organisms at deep sea hydrothermal vents. These things are 2,200 meters under the surface of the ocean, so no light from the surface whatsoever penetrates that deep. The light from the surface goes only 100 or 200 meters down, and then it's gone. So it's inky black down here, except these things are hot, so they give out black body emission. I'll show you the curve of that in just a minute. And Cindy Van Dover, who was a vent biologist, had the idea that because of this light emission that's given out by these vents, that there may actually be organisms that can use that light to do photosynthesis. And so she got me involved in this, and we went down and had some...
One question.
Yeah.
That was you on the submarine?
That is me getting into the album. It's not a very good picture. It shows up much better on my laptop here. Yeah, that's me getting in the album, and that's another whole story itself. It's an 8-hour trip from surface to surface. 3 people in a 6-foot diameter sphere with no restroom. It's quite an experience. It's really an experience of a lifetime. And so I can tell you about that over a beer at some point. So it turns out that Cindy's ideas were correct, and we actually did discover an organism that lived down there. And this is a culture of it, and these are some micrographs of it. And it's a new species of bacteria, and it turns out it contains these chlorosomes that I've been telling you about. And it uses as light source, or as an energy source, the infrared tail, near IR tail of the black body emission from the vent. And this shows just a black body emission curve from the vent. It's about 300 to 350 degrees C. So most of the intensity of it is peaked out in mid-IR. It's just a tiny tail that comes down into the visible and near infrared region. And this is the region where these chlorophylls in these chlorosome complexes are formed. So they're scrounging for very low amounts of light. You can actually visualize it not with the naked eye, but with an infrared camera or an image intensified camera. You can see the light coming out of the vents. But it's sort of a remarkable thing that these organisms can actually live off this light that comes out of the vent. And these organisms are obligate photoautotrophs. They have to grow using photosynthesis, and so they probably are indeed using this light in the vents.
What were the dots on the curve?
Oh, the dots are the actual measurements.
Oh,
So these are the real light measurements that were made. And
why does it differ so much from black body?
well, there's a second source of light that is not black body emission, but is thought to be something like triboluminescence or crystalluminescences. These things, the vent fluid hits the seawater, it precipitates, you get all that sort of black smoke appearing, and there's actually luminescence, very weak luminescence that comes out from that. Probably our organisms are utilizing more of this black body part of the curve. But it's and the precise origin of this shorter wavelength light is really not terribly well understood. So hopefully that kind of whets your appetite that these might be interesting things to study. And the organization of the pigments in these systems has been something we've studied in my group for a long time, in not those vent organisms, but relatives that are found in the more surface environments. And it turns out they have very little protein involved in them. So in contrast to the pigment proteins that I showed you in all those other systems, where the chlorophyll molecules are actually oriented by their association with the protein, here you have a chlorophyll-chlorophyll, or pigment-pigment interactions that form these sorts of oligomers and into higher order structures here. And they're particular chlorophylls. These are not the standard chlorophylls that you find in other systems. In fact, they're only found in these chlorosome complexes, and they have slight structural variants from the normal chlorophylls. In particular, this hydroxyl group here serves to coordinate adjacent molecules, and they lack a bulky substituent here in this position, so that the rings are able to pack much more efficiently. And we don't yet know exactly what the structure of these complexes are here. These are three of the earlier ideas, and I'll show you some more in a minute. But one thing that's interesting about these things, they will exhibit reversible self-assembly. And they're similar to J aggregates, and you get this very strong coupling of the transition dipole moments of the chlorophyll molecules. You see a large redshift. The monomer spectrum is about here, about 660. See, it shifts out to about 750 or so nanometers, similar to what's seen in the J aggregates that are found in photographic dyes. And that will be, you can modulate that, you can break that up, break up that assembly, and the absorption will shift back to the monomeric form, and then if you permit it to reassemble, it'll then immediately go back and reassemble this complex, and you can do that again and again. So it's a remarkable sort of self-assembling system. It has some appeal in terms of a possible artificial system. Saw some of these rod structures that are there, and I'm not going to go into this in any detail. There's been a lot of effort on a variety of techniques. This particular paper is a recent PNAS paper from a Japanese group, not our work, but using 2D NMR, solid-state NMR, I should say, to try to understand the larger scale structure of these things. And there have been some evidence from EM work that you have these large tubes that are basically pigment oligomers, and then they propose a sort of a pathway of energy migration here. However, this has recently been challenged, and that rod model would predict a particular behavior in a small angle X-ray scattering experiment. And Jakob Censik and his colleagues from Czech Republic have gone in and measured that, and this is the solid line here is the measured spectrum, small angle spectrum, and as you can see, there's absolutely no feature whatsoever that corresponds to what is predicted for that rod. And in fact, there's a much smaller scale feature. This rod is supposed to be about 10 nanometers in diameter, and they don't see anything that looks even remotely like that. And so this has become sort of a controversial thing in the field. They've produced another model that represents, that incorporates some of these experimentally derived features there, and this is called the Lamellar model. And so instead of having these tubes that run the length of the complex, they propose these sort of undulating lamellae that contain the aggregated pigments. And the sort of local structure of these is probably fairly similar in all the models, in that you get that face-to-face association of the chlorophylls, but the larger scale structure is still quite a controversial thing in the field. We just started a collaboration with Censik to try to look at some other species that we have that have somewhat different types of chlorosomes and see whether or not we see that same sort of behavior with them. So that's an area that is still quite an active research area. Now, if you go back to there's another antenna complex here that's called this FMO complex, or bacterial chlorophyll A protein. It's a trimeric protein complex that sort of serves as an intermediary in the energy transfer. So here's this large chlorosome with hundreds of thousands of pigments organized in terms of these oligomers, like I've been showing you. Then there's another intermediate, smaller protein complex called baseplate complex, and then that feeds energy into this FMO complex, which ultimately feeds the reaction stream. So FMO is a very interesting system and has been studied a lot over the years. Structure, the FMO refers to Fenna, Matthews, and Olson. Fenna and Matthews are X-ray crystallographers. It's Brian Matthews from Oregon who first determined the structure of the protein that John Olson had first purified back in the 60s. And it's a trimeric complex, and it has, each one of these monomers has 7 molecules of bacterial chlorophyll in it that are wrapped up. Remember we had those taco shell things for lunch. These proteins actually are remarkably similar to that sort of taco shell shape. In that you've got the beta sheet structure, which sort of forms a taco shell, and then the protein, or the pigment in the middle is kind of like the stuffing, and then there's a little bit of lettuce on the top of the alpha helical. But this has been a very, very interesting system to understand, because it was the first chlorophyll protein complex to have its X-ray structure determined back in the 70s. And so a lot of what we know about how chlorophylls interact with proteins and interact with each other was determined from analysis of both experimental and theoretical analysis of this complex. And so we've continued to work on this system for a long time, and it still has a lot of interesting things to reveal to us. And some of you may have seen this very interesting paper that we had a collaboration with Graham Fleming's group from Berkeley, in which we did 2D ultrafast spectroscopy on this FMO complex, and this shows the diagonal, which is basically similar to just the absorption spectrum. This is low temperature absorption spectrum. Each of those 7 bacterial chlorophylls in that taco shell environment has a slightly different absorption property, and then as time goes on, you can develop off-diagonal elements very analogous to what you see in terms of 2D NMR. And here the off-diagonal elements develop because of electronic coupling rather than spin coupling, which is what you would see in the NMR case. And from that, you can determine, these are now the geometries of the pigments, and you can, from that off-diagonal coupling, you can determine that there are at least two different pathways of energy transfer within the complex. This represents the energy level diagram, and you actually have the lowest energy emitter here is pigment number 3, and that one can be populated in a couple of different ways. And we have a new paper that's just coming out in Nature the next month or so, in which they've done some very high time resolution experiments with a lot of very interesting quantum oscillatory behavior. How much time do I have, Tom?
About 10 minutes.
10 minutes? Okay, that's just about the right amount of time. Here I've found a picture of a knob on the internet and superimposed it on this complex. And that's supposed to be a metaphor for a volume knob to control the regulation of the energy that flows through this system. So this is, in a way, how do you regulate this system? You've got these 200,000 pigments here feeding energy in through this complex system to this reaction center complex, which has to process it in terms of the electron transfer processes. And so you can imagine that if this thing gets under conditions where there's too much light that saturates the photochemistry here, or if for some reason the photochemistry is not working properly here, that on this molecular scale, that sort of energy input will very rapidly lead to very strong photo damage in these systems. And so what's found in all photosynthetic antenna complexes, but here it's perhaps more developed than in others, that you have multiple regulatory and repair processes that go on to first control the amount of light that energy that gets delivered to the reaction center, and then if there is photo damage, to come in and repair it later. I think these, again, are things that we're going to have to understand, these kinds of regulatory processes, if we're ever going to build artificial systems based on these sorts of principles. And so this is very highly regulated to prevent over-excitation and damage, and in particular damage if there's any oxygen present, because these particular organisms are quite anaerobic organisms. And one of the things I was telling you about this FMO protein, the trimeric protein, it has some very interesting excited state relaxation pathways, and this is just a time-correlated single photon counting lifetime study of these organisms, of these isolated complexes, and it shows that the dominant decay pathway here is about 50 picoseconds under sort of ambient redox conditions, and I couldn't find the slide, but if you then add a reductant to get rid of all the oxygen or in some way make these systems completely anaerobic, what you find is that very short lifetime decay process is replaced by the dominant lifetime now being about 2 nanoseconds. It represents the decay of just an isolated antenna system. So what's happening here is that there's some sort of redox-regulated quenching process that happens within this purified protein. And we don't yet know the molecular nature of that, but we're working with Matthews's group from Oregon to get an ultra-high resolution X-ray structure, because there are some spots in that structure that have anomalous electron density. We think there's some redox-active species in there that hasn't been resolved in the previous studies. And so there's one layer of that process that goes on. There's also a very strong quenching process that can happen within the chlorosome complex itself, those aggregated pigments that I showed you earlier. And here this is perhaps a little bit complicated, but if you compare the absorption spectrum, solid line, to the fluorescence excitation spectrum in dashed line, and you normalize it with respect to the direct excitation of the long wavelength pigment that is fluorescing. When you have a sensitized fluorescence process, you can do this. The amplitude of the fluorescence excitation spectrum compared to the absorption really gives you a direct measurement of the energy transfer efficiency. So under anaerobic conditions, these things have 90-some% efficiency of energy transfer. So essentially every photon that's absorbed in that large chlorosome complex is transferred down to the reaction center. So they're 100% efficient in that respect. If If you let the samples become slightly aerobic, and we've actually then gone in and done redox titrations of this, you see that this fluorescence excitation is much, much weaker. And in this case, it shows about 20% energy transfer efficiency. Actually, under most conditions, it's down in the 5 to 10% condition. And this can be modulated back and forth. And again, that turns out to be a redox process. That here's another way of measuring that same process. Here's now looking at cytochrome oxidation, but then as a function of light intensity. And this shows that under the anaerobic condition, you have very efficient light intensity. It doesn't take much, very efficient process. It doesn't take much light to saturate it, but under the aerobic conditions, it's very inefficient. And so this represents a very strong quenching of excitations in this antenna. And in this case, we understand the molecular mechanism of it. These complexes actually contain unique quinone called chlorobium quinone, which is only found in this chlorosome complex. It's the only place in biology that it's found. It's a naphthoquinone derivative. And what it does in its oxidized form, it undergoes reversible electron transfer quenching with the excited chlorophyll. In the reduced form, which is the form it would be in normally under the efficient growth conditions, it would not accept an electron, and so it would not be a quencher. And so we see this very dramatic effect here. And so this just sort of summarizes some of these regulation processes, and there are at least 3 separate levels of this. This last one I was mentioning involves this chlorobium quinone, which can directly quench the excited states in the chlorosome. This FMO protein has some as yet unknown redox active group in it that will also, under certain conditions, quench the excitations. And there's a third process, which I didn't have time to talk about, in the reaction center, which is yet another layer of this. And so there's this multiple layer process in terms of regulating the energy transfer in these systems. So that brings me to the end. I just want to point out people who've done this work. This is my current research group, and the students who are involved in this work. Jianzhong Lin has done some of the FMO work that I mentioned, and Aaron Collins is doing work on the chlorosomes, and then other people are working on various other sorts of projects that I didn't really have a chance to talk about. And then over the years, there have been a lot of people in my group who've worked on these systems, and I want to put at the head of the list Gabe Montano, who's here in the audience. The student who worked with me several years ago and did some beautiful work on determining the number of pigments in the chlorosome and also isolating and characterizing that base plate complex. And so these are various students. First group here are students. These are postdocs, and then some staff members and technicians here. Kind of collaborators with a number of people. I mentioned Fleming's group. The Photosystem I group was done. Work was done in collaboration with Jim Barber's group from Imperial College. The work on the hydrothermal vents was done in collaboration with Cindy Van Dover and Tom Beatty and Jorg Overman and Jerry Plumley. And then some of the structural work on the FMO was done in collaboration with Jim Allen and also Brian Matthews' group from Oregon. And EM work was done with Robbie Robertson at ASU, and some other people here have also been involved in this. And we've had support. The Photosystem I work was supported by NSF Physics Program, and the DOE has been a long-term supporter of our work on the chlorosome and other antenna systems. So thank you for your attention.
Questions?
How efficient are these biosystems, for example, the react?
Well, the efficiency is a kind of a fuzzy word because there are different levels of efficiency. If you look at the quantum efficiency of photochemistry in the isolated reaction center complexes, people have measured that. And under low intensity conditions, that's basically 100%. So every photon that's absorbed leads to separated charge. So that's sort of the highest efficiency. If you look at it in terms of the overall solar energy conversion efficiency, like you would comparing it to a photovoltaic, it's down in the few percent range. So you're not collecting, in terms of stored energy, you're not collecting a very large fraction of the total energy in the sunlight. Of course, that represents a lot of light that the organism didn't absorb, that was in the near infrared that it didn't see, and so on. So I think it's fair to say that in almost all cases the natural systems don't approach the level of energy efficiency that you would get in something like a modern photovoltaic device. So if that's the sort of question you're asking. But there are lots of different ways to get at that question of efficiency, and so you have to be very careful that the question is framed in a way that isn't misleading, because people will sometimes tout, oh, these things are 100% efficient. Well, they might be with one specified definition of efficiency, the photochemical quantum efficiency. But in terms of the overall light intensity conversion, they tend to be much less.
So given the fact that each chromophore, each pigment has its own range of absorption, if you were to design a biomimetic system and were to mix and match different pigments, will you be able then to increase the collection efficiency in that?
Yeah. The existing organisms have done a pretty good job of filling in some of those holes in the spectrum. Chlorophyll absorption, of course, has very strong absorption in the red and in the blue region, not so strong in the green, but there are carotenoid pigments and other pigments that fill in that region, such that if you actually look at the reflectance spectrum of, say, a leaf from a tree, even though it obviously looks green to us, the amount of that green light that's being reflected is a very small fraction of the total incident green. So it uses even most of the green, uses all of the red and all of the blue. So actually, I'm working on a paper that's titled Plants Are Not Green, They're Black. And so plants, in a certain sense, really are black in terms of they basically absorb and utilize all the light that hits them. I mean, obviously with a small amount of that green that is not utilized. But they do that with other pigments, and I didn't really dwell on that, carotenoids and violin-type pigments and so on that fill in some of these gaps in the middle. They don't utilize all the energy of those shorter wavelength pigments. In fact, what they do is there's a very rapid energy transfer down to the lowest excited state level of the chlorophyll, depending on the species, down to around 700 nanometer, about 1.8 EVs. So even that blue photon, most of that energy in the blue photon is lost as heat, and it degrades down to the level of a red photon very quickly. And that's the energy that's available for photons.
Also on those lines, have you yourself or are you aware of anybody who has performed site-directed mutagenesis in an effort to alter the molecule, the protein structure?
There's been lots of that. There's been lots of that kind of work done in all of these systems for various ends. I mean, every one of these systems, well, not everyone, but most of them have genetic systems available such that you can get in and make site-directed changes to the protein and look at the consequences of that. So there's a lot of that kind of stuff that's been done. I mean, it's a big area. One here and then.
Yeah, this is a little off your main topic, but you mentioned that Photosystem II has now got a well-characterized structure. Does that include the manganese complex, or is that still up in the air?
Well, the manganese complex has been quite controversial, as you might imagine. That's kind of the heart of where the oxygen is produced, and that's really the tough chemical step in all these systems. And there are a couple of X-ray structures out there that include the manganese at relatively low resolution. There's a very nice paper that came out about November or so in Science by Junko Yano and Ken Sauer's group at Berkeley and Vidali Chandra, in which they've taken the X-ray data and XAFS data and sort of put it all together in polarized XAFS and have produced what I think is at least now the best reliable structure for that manganese complex. And so it's not a solved problem, but they've made a lot of progress on it. And I think if you're interested in the latest on that subject, find that paper that was out recently in Science, because it's a very nice paper.
Thanks.
Andy?
So Bob, if you just look at a big aggregate of chromophores, at first glance it's not really obvious what kind of genetic control that is under. And I wonder what's known about how these aggregate systems have been evolutionarily optimized.
Yeah, that's a really interesting question. And I sort of emphasized the fact that the proteins are not involved in the pigment organization to form these large aggregates. There are proteins in that complex, that chlorosome complex. They're around the envelope of it. And somehow they are involved in defining the scale, the size of this complex. And then, of course, the pigments have to be biosynthesized, which happens on, there's a complex biosynthetic pathway for the pigment synthesis, and then inserted into that chlorosome where they then, I think, will self-assemble into these structures. So they kind of form a framework or a scaffold that you put these pigments in, and then they will self-assemble into these structures. But there's a lot of genetic control involved in that. Some of the organisms are very highly regulated in that they will express these chlorosomes only under certain growth conditions and not under others. And in most cases, they will respond to light intensity. And if you go to very low light intensity, they'll make the complexes larger and make more of them and so on. So there is regulation on that sort of more genetic scale of it. But in terms of the actual biogenesis of these things, we had a paper that just came out a few weeks ago in Febb's Letters. A student of mine, Martin Holman Marriott, proposed a new mechanism for how these complexes will originate. Where do they come from? I mean, they don't divide like a cell does, and so he has a model that he's proposed for how these chlorosome complexes can sort of originate.
Are there any thoughts as to why these kinds of aggregate types of assemblies aren't used more often? I mean, you might think they're somehow, I mean, naive.
Yeah, they're cast me out as far as I'm concerned.
What's that?
Yeah, that's a good question, and I can't really say. They seem to work really well when they're perfect, but if they get damaged, they can perhaps not be the best. But that's a complicated question that I can't really give too much insight into. You tend to find them only in two groups of the anoxygenic bacteria. You don't find anything like this in any of the higher plants or oxygen evolving systems, and so they seem to be a system that developed fairly early, and it's a simple system in the sense that probably doesn't need too many genes to code for these things, unlike some of these more sophisticated systems that you have in higher plants. And so maybe Mother Nature hit on this fairly early, and then it was replaced in these other complexes. But I can't really give a very deep answer to that question.
Maybe one last question. Paul?
Well, I was just wondering whether resistance to superoxide is a factor in this, because I just say just learning that a lot of photosynthesis issues in dealing with some very highly oxidized.
Yeah, I think that's undoubtedly important in these organisms where I was showing you the oxygen regulation of these things. And I think that it really does ultimately come down to superoxide in that case. And what happens in those particular organisms is the reaction center produces a very highly reducing species in terms of ferredoxin. And that ferredoxin is then free to diffuse through the cell. If there's oxygen there, it's very readily auto-oxidized to form superoxide. And so you don't find that really diffusing ferredoxin present in other systems. And so in this particular case, the cell is, in a sense, very vulnerable to superoxide. And so I think that avoiding the production of superoxide is, in a sense, the point of several of these regulatory processes. So I completely agree.
Okay. I guess as this morning we'll take a break now after the second talk, and so maybe we could reconvene at 3 quarter after 3.